Assalamu alaikum. Good morning, good afternoon, good evening, everyone. Thank you for joining for almost our last webinar for the year. Inshallah, we have a whole list for next year, which I will share soon. But today I would like to welcome Dr. Naweed Syed. I don't have words to share how amazing and how great he is. I have always admired him. But I will give that opportunity to Dr. Shabih. I think he can do a better job than I am. So, Dr. Shabih, please.
Well, thank you, Sabrina. It's not possible to justify. I mean, I was looking at the Wikipedia. It's in about 10 pages. I mean, Naveed's contribution to science is beyond anybody else I know in our fraternity, family, friends, organization, IMI, CV, anywhere. He's top of the list. But anyway, I'll just go to a few points I've made. No, Naweed Syed is a famous Pakistani-born Canadian neuroscientist. He is the first scientist in the world to connect the brain cells to a silicon chip, creating the world's first neurochip. I remember distinctly many years ago when he came to London and gave a talk. Everybody was bamboozled. I mean, that was an amazing work. And in those days, we used to talk about Steve Hawkins and all these people. But of course, in later days, Elon Musk came over, and Naveed will tell us more about it. Naweed is a professor at the Cummings School of Medicine, University of Calgary in Canada. He has huge number of papers, and not published in ordinary journals, but journals like Nature. I mean, Nature is number one in the world. Everybody knows that, those who are interested in research. His focus of research is cellular, molecular biology, and more intensely into neurosciences, and his work is published so extensively. I'm looking at that list of journals, amazing work that he's done. But more important than that, Naweed is our friend, and he is a strong member of Miami Medics International. Also, he's a very strong faculty of Sadiq International Virtual University. Welcome, Naweed, and over to you. So we'll do interactive. We'll have people like Mushir and Mulazim Bukhari and Syed Kazmi. We can have like welcome podcast, if you don't mind. So we can start with the work, and this is a fascinating subject. Being a surgeon, I noticed in doing as I was saying earlier on, some of my nieces were senior. They used to have these problems. We'll talk about that. Over to you, Naweed.
Thank you, Shabih Bhai. As usual, I think the only pride that I pride myself is that associated, affiliated, and I know you and your Shafa Ziyarat. Also, I think it is the credential that I really go by. And somebody had asked Imam Ali alayhi salam, what do you consider to be the most important attributes of your personality? And he never mentioned Khaybar Khandak or anything. And he said, I take enormous pride in the fact that I stitched Prophet, peace be upon him, his shoes. So I think, that is really the credential. But I also really owe a huge debt of gratitude to you and your entire team for spreading the knowledge in different parts. I think today, normally we talk about I've given talks about Alzheimer's disease or brain connectivity, neurodevelopmental disorders, and also about brain chip technology. But I think one of the topic that is really coming up is people are noticing that children who are exposed to anesthetics, even for 30 minutes or for multiple times during early childhood, they end up having cognitive decline and learning and memory problems. Similarly, when you look at the elderly population, and it's usually after perioperative neurocognitive disorders, 50% of these patients, elderly especially, they have prolonged hospital stay, they have loss of independence and increased death. So I think there is something, an issue that pertains to anesthetics that we needed to really explore as to what is really happening. So this will be really the topic today, is that general anesthetics. Of course, we know that they are necessary for all surgeries, millions of them that are performed around the world. But we don't really know the exact mode of action as to how they act and what could be the long-term impact of exposure to young children and also even pregnant mother. So I won't go into the nitty-gritty details. We have published several dozens of papers which compelled our Canada Health Agency to force that the labels be put on general anesthetics, especially when they're giving to young children or pregnant mother, that there is a warning when it's giving. But it's something, it's like you can't perform surgery without anesthetics. So people say, well, we don't really have a choice. So I think hopefully today we might be able to come up with some alternative strategies, which could be the enrichment. So it's a good morning from my side, and good evening, good afternoon, good night, wherever around the world you are. And I think this particular organization, that the way it organizes these particular webinars or seminars, it's really, really very, very critical, and especially for our people who do not normally have access to cutting edge research. So Calgary is located in the foothills of Canadian Rockies. We are only half an hour away from the mountains. It's a beautiful city. Right now, a little bit snowed. And so what the learning objective I was trying to achieve today is that how brain circuits are really put together during development and communicate with each other through synapses. As you know, that there are tens of billions of brain cells, more than what you see in the Milky Way. And there are trillions of synaptic connections between these brain cells through which they communicate with each other. And this is all put together during early development. So this is one of the area that I will highlight how these circuits are constructed. This is one of the area of my research, and we have published several papers in Science, Nature, and other journals deciphering fundamental mechanisms by which brain cells are connected in the intact brain during development, and to give rise to behavior, cognitive functions, learning, memory, and really day-to-day tasks. Then we will talk about how might various anesthetics affect neuronal communications, and they impact the cellular viability, meaning they cause a lot of cell death, and then they alter brain cell connectivity, so they affect the growth of the brain cells, and they also affect various synaptic connections that form the basis for learning and memory. So the question I will also pose today is that do anesthetics that perturb learning and memory, and in so doing, do they invoke cellular, molecular, and genetic mechanisms? And I will highlight some of the genes that are altered in an animal model when they are exposed even once as neonates, but when they are tested in the adults. So the question is, how can one then mitigate this anesthetic-induced toxicity, as anesthetics are really pivotal, essential for our surgeries, and surgeries cannot be performed without them. So we know that the brain is the fastest growing organ in a human body. And you can see from here, all the way from this is the brain, and as compared to all other organs, it is the fastest growing organ. And when a baby is born, the baby actually has twice the number of brain cells, but soon after birth, half of them are killed because of natural programmed cell death. And after that, there will be no addition of new neurons in the human brain once all the synaptic connectivity is established. But these synaptic connections in the brain are really the fastest growing, and as a result, anything that will impact their mechanisms by which they recognize will alter the brain for a longer term. And this is important, especially in the cases of mothers who are undergoing, pregnant mothers who are undergoing any kind of surgery during the late stages of pregnancy, that anesthetics given them cause major problem. And an interesting thing that has just come up as well is that normally in the brain you have GABA, and the GABA, which is targeted by a large number of anesthetics, is normally in the adult brain is inhibitory, whereas glutamate is the excitatory. But what happens is during development, most of the connections that involve GABA receptors, they turn out to be excitatory. So now, even after surgery, people are looking at the overexpression of these alpha 5 GABA AA receptors, which they think is really the culprit for the cognitive decline or the delirium in the older population. We'll talk about this a little bit later. So you can see that as compared to other organs, for example, stomach, to the brain is the fastest growing organ. I mean, mind you, what happens after is that when you grow up and you become either a professor or a politician, I think our brain tend to shrink, but the stomach gets bigger. Just a pun. So there are genetic and environmental factors that will change our brain circuits, all the time and throughout life. Well, one important takeaway is that brain cells or neurons that fire together, they wire together. So I think this activity pattern is essential for brain cells connectivity. And then neurons that fire apart, they fall apart. And this is what happens in the elderly when all of a sudden, or even in Alzheimer's disease, when these brain cells are not firing together, they end up really falling apart. So neurons that are out of sync, they fail to link. So I'll give you an example that if you have a kitty cat or a duckling, and you put a patch over one of its eye just for a few days during critical time period when brain cells are connecting through activity dependent mechanisms, what happens in that case is that that kitty cat will be blind in that eye for life. And similarly, children who can't hear, they can't speak, because these centers are really interlinked and intertwined with each other. So what it really tells us that the cells that fire together, they wire together. If they don't fire together, they fall apart. So the connectivity never really happens. So this is important for the rest of the talk, because anesthetics, what they do, they block this activity. It will be just like putting a patch over one eye of a kitty cat or a duckling, and then studying them later on in their life. And it's a very well established fact that if you don't have sensory input coming from your retinal ganglion cells, it will not connect with your individual cortex. So this is something really important, is that anesthetics will block this activity pattern. So one drawback will be that the synapses will not form. As a result, those circuits that were ascribed to a particular behavior, they will not be in place for you to invoke them. So this is one thing. And also, I think this activity, you use it or you lose it, but those elderly patients who are undergoing cognitive decline, already, if they undergo surgery, major surgery, hip replacement, a knee, and you block the activity in their brain, the synapses are so vulnerable that they will fall apart. So this is what we see in perioperative neurocognitive disorder, which is generally seen in 50% of the elderly patients. So this is really the takeaway of this talk. And if you have stayed awake, probably you can go to sleep now. But what happens is that during only, excuse me, only a brain somewhat organized can sort out what's really worth paying attention to. So from early development to newborn, many factors will then contribute the shaping and the connectivity of the brain circuits. If your brain is exposed to an enriched environment, for example, when we have these little rats where mama licks his pups, those pups are good learners as compared to mother who doesn't lick its pup or pays much attention. If you switch them around, they become either good or bad learners. So I think we know that the enrichment plays a very important role. If a child is even during pregnancy, mother is exposed to stress and other factors, abuse, neglect. Those babies will have perturbed or altered brain circuits. So babies actually don't know what they would need for their lives, so they pay attention to just about everything. So in a family where both parents talk about their work, they talk about their daily activities, what's going on around in the family, what's going on in the technology development sector, in community, and in society in general, those children will have a 3.5 million word vocabulary, greater than children where parents do not talk to their children. So child's brain is just like a sponge. It takes up everything. And because they don't know what will be important. And in the Quran, Allah says also that when you were born, you were completely clueless and ignorant. We gave you ears and also eyes to hear and see. And this forms the basis for your cognitive functions and learning and memory, and eventually defines you who you really become. So it's really important that early enrichment is very, very important. So our research program, we have come a long way to deciphering individual brain cells and how they connect with each other, how this connectivity elicits a particular behavior. But these behaviors that we have, or the network connectivity that we have, is not hardwired. It's actually synaptic plasticity that allows these circuits to change all the time. So even after this particular session, if you paid attention, you would have learned something which may form the basis of new synaptic connections. And if these synaptic connections are enriched, probed, explored, and really exploited, you will get synaptic plasticity, which will alter the behavior in the longer run. So we know that there are epigenetic factors that are important. And we also know that the genes define the basic neuronal circuits that are really form the basis for some connectivity where Mother Nature doesn't want to take any risk. So look at this experiment.
Humans in this experiment have been raised in isolation. They've never seen any other birds.
And there I see is our friend the goose. There wasn't any reaction at all, was there?
No, there wasn't. That's what interested Professor Melzak. You're seeing what happened when he first did this experiment in the London Zoo. If you were a duck, would you worry if you saw a goose?
You mean he will worry when Professor Melzak changes that goose into a hawk?
Well, let's watch.
Really frightened duck.
Well, not all ducks react this way. Some are simply wary when they see their first hawk.
So in real essence, you could see that some circuits in your brain are hardwired. And we see also in a deer, when a deer is hit by floodlights, it just freezes because it knows that any movement will get the predator coming after him. It freezes as if it doesn't really exist. And pigeons, when they see cat, they oftentimes close their eyes because they can't see the potential threat. So these are really hardwired circuits into our brain. But everything else that you see is really, contingent upon nurture. So these are the brain scans of 2-3 years old. One is the normal child to the left, and one that was subjected to extreme neglect throughout their lives. So you could see not only the brain size shrinks considerably, but also you can see that there are huge gaps in the connectivity patterns. So the takeaway from this particular really slide and then a couple of slides is that sensory inputs or the nurture aspects or the epigenetics play a very important role. And this is all contingent upon sensory drive that's coming into the brain. So the brain cells that will fire together, they end up wiring together. And if they fire apart, they will fall apart. So this is really something that's really important. So how brain cells really communicate with each other, we know that in many cases,
is a crucial junction between two neurons that allows them to communicate without being physically connected. A synapse has several structures. The presynaptic neuron ends in a small bulb called the presynaptic terminal. The postsynaptic terminal membrane is directly adjacent, separated by a small space called the synaptic cleft. An action potential causes voltage-gated calcium ion channels to open in the presynaptic terminal. The influx of calcium ions prompts synaptic vesicles to release neurotransmitters via exocytosis. This neuron is releasing acetylcholine. The neurotransmitter quickly diffuses across the synaptic cleft. Two acetylcholine molecules bind with one ligand-gated sodium channel at the postsynaptic membrane, opening the channel. When enough sodium ion channels open, the postsynaptic cell depolarizes and the action potential continues along the neuron.
so in a nutshell, what you really have is you have a presynaptic cell, which is doing the talking. In this case, if I am talking, I'm the presynapse. You are listening, you're receiving this information. You are postsynapse, and this internet is acting as a synapse, synaptic connection between us. So when you look at the presynaptic action potential, as I said, whenever this presynaptic action potential reaches the terminal, it opens up voltage-gated calcium channels. Calcium enters in the terminal. It binds to the vesicles. Vesicles will then really release in the synaptic cleft and then activate sodium, sodium channels, which is normally, gated with glutamatergic synapses, open up, and then the information transfer from presynapse to postsynapse. So there are two important proteins that are really important. One is synaptophysin, which is a marker for presynaptic cell. And then you have postsynaptic density protein, PSD95, which brings these receptors on the postsynaptic side, tethers them, and then regulates their expression, whether they need to be more of them or more of them need to be phosphorylated. So this is really how the synapse functions. So the neurotransmitter glutamate is the main excitatory neurotransmitter in the CNS, because GABA brings about most inhibition. Other transmitters, for example, dopamine, serotonin, acetylcholine, and many other peptides are also functional in our brain. It's really important to know that most drugs and anesthetics target these specific receptors and the ion channels to bring about the state of unconsciousness or anesthesia. So they shut down the excitatory synaptic transmission, but they augment inhibitory synaptic transmission. So what happens after injury or surgery or even early developing brain is that these GABA receptors tend to be excitatory in nature. Which it does is that actually it is a counterintuitive to any of the effects that you would normally see under normal conditions. So as we know that the pain really changes people, so you can't perform surgeries without anesthetics, and then local analgesics are given to manage pain, and there are many other narcotics. So pain of mind is really worse than the pain of body. We also know that people may have psychological pain, emotional pain, phantom limb pain. All of these, the pain changes people. It makes them trust less, overthink more, and shut people out. So people really get isolated. So time doesn't heal anything. It just teaches us how to live with the pain. So the greatest evil is the physical pain, as Saint Augustine said. All surgeries and pain management, strategies require anesthetics, be that general or local. So we will now, from here onward, explore one particular anesthetic agent that is used generally in a developing children, young brain, which is sevoflurane. So the state of anesthesia and pain control really require transient suppression or blockade of neuronal activity and synaptic connectivity. So the modes of actions and the potential target sites of anesthetics are often difficult to decipher in the intact brain because we don't really know where they act. Another interesting aspect is, which has led to really many lawsuits, and also in the clinical, in the field of surgery, is that some people, even though the anesthetist may think the person is anesthetized, but they are still awake. And they can really listen, they can feel even the pain, but because their body is paralyzed, they cannot really react to it. And this has led to litigation. So the question really is, do all synaptic connections are influenced by anesthetics the same way as we see in normal people? And what we also know is there a major mutation in a sodium channel. For example, a child will walk into an intensive care or emergency, especially in the emergency department, and his hand is dangling and he has no clue what happened, feels no pain. And you see in Pakistan, I think there are families where their entire genome was cloned, and these kids born to those parents, they could not feel any pain whatsoever. They will take a sword from one side of the mouth and put it the other, and they have no really pain. And they don't live very long because then their injuries really exacerbate and cut short on their lives. So I won't go into too many details of papers because I think most of the work is already published. You can just search our names and you will find we were the first team to actually demonstrate that the anesthetic affect not only synaptic connectivity, cellular viability, their growth, and connectivity in several papers. And a comprehensive review is listed here, which was published just a few years ago in Journal of Neurophysiology, where we highlighted most of these studies. And we recently published a couple of papers showing that sevoflurane exposure, when you take these neonates, young pups, newborn pups, and you expose them to sevoflurane just for 30 minutes or so, then you allow them to become adults, and then you test their learning and memory. Most aspects of learning and memory are okay, but they falter in two major areas, which I'll talk about. So when you look at the actual synaptic transmission between brain cells, an interesting thing is every single action potential will trigger an excitatory postsynaptic potential in its connected cells. And you can see that most of these excitatory postsynaptic potentials that are subthreshold, they don't lead into generating action potentials. But then when you fire a tetanic burst, you get much bigger response than individual EPSPs. But if you come now and fire a single action potential, you can see that the EPSP after post-tetanus is significantly really enlarged, and you can see it's augmented. So this is short-term potentiation. We call it working memory or synaptic plasticity. And the interesting thing is that it is actually not time dependent, it's use dependent. So if you do not fire this action potential for 5 hours, whenever you come back and fire this action potential, the synapse will still be potentiated. So this really shows you that the synapse get loaded like a gun, and they are waiting for somebody to pull the trigger, and they are all released. So we define this as PTP, post-tetanic potentiation, short-term plasticity. And this is what happens is that It's an interesting topic in its own right, how we humans have really given up on this short-term memory, short-term potentiation at the expense of acquiring languages. So we asked the first question, and I actually did this experiment. My own daughter, she had a minor surgery. They had to put her to sleep, and the anesthetist came, talked to her, and I asked afterwards, what was the accent of the anesthetist? And she said he was British gentleman. So that really told me that her short-term working memory, she had never met that anesthetist. She had no clue that I will be testing her. And so we wanted to test this in a real sense, that what actually happens is when you fire first action potential, you have these vesicles that are released. As I showed you, they bind to the acetylcholine receptors, and then you get synaptic communication from cell A to B. But if you fire tetanus, a lot more calcium comes in. It will mobilize more vesicles, but it also activates calcium calmodulin dependent enzyme, and it brings those vesicles to dock at the synaptic site. When you come back after tetanus, you fire single action potential, those vesicles are released at mass, and you get that potentiated effect. So we asked the question, we know that the anesthetics question, do they affect brain connectivity and function? So the general really hypothesis is that the anesthetic induced perturbation of neuronal activity and function renders the brain connectivity dysfunctional. And so to test this, of course, you can't do this in the intact animal or human brain, or even in the intact animal. We decided to use a different approach of isolating these brain cells from a rat or mice and grow them in cell culture, and ask the question, when they are grown, what happens to, if they are exposed to anesthetic soon after plating in a culture dish, what happens to their growth, and what happens to their really connectivity and formation of synapses. So we have markers of all kinds. We have presynaptic markers for synaptophysin, postsynaptic markers for PSD95, and many other proteins that we can target and then localize and see what happens. We can also use patch clamp techniques, whereby we can grow these brain cells either on a chip and then patch clamp them, record ionic currents. So the first question we ask, do inhalation anesthetic affect short-term plasticity, underlying working, and memory? So I had shown you already that when you fire single action potential, you get EPSP, you fire tetanus, you get compound PSP, and if you come back and fire the same action potential afterwards, what actually happens, you get an augmented response. So what we did is we exposed these synapses both before, during, and after testing of this synaptic plasticity. And what we normally see is the synapses potentiated several hundred times after the tetanus. So you can see that this potentiation, and then it declines and comes back to baseline. So we exposed them to 1.5% sevoflurane. And what we found, that even though the synaptic transmission was suppressed, but really the ratio of PTP from here to here and then here to here did not change much. So it really shows that the sevoflurane does not affect, when used in clinically relevant concentration doses, does not affect short-term plasticity. So this was really a big relief that, it's okay. And even after exposing these synapses for several minutes, we still find that the sevoflurane does not affect synaptic potentiation. Even after several minutes to hours, the synapse will always be potentiated, just like in a controlled situation. So the sevoflurane will do the same thing. So what we wanted to do was that asks 3 simple questions. Does anesthetic exposure affect, sevoflurane particularly affect, induces cytotoxicity, which will be cell death? We will look at the extent of growth and also synapse formation. So 3 different facts. So what we do, we have these pregnant mothers, P0, Sprague Dawley pregnant mothers. When the pups are born day 0, here we take these pups. We will normally extract their either hippocampus or cortical neurons, and then we grow them in primary cultures. And then in so doing, what we will do is earlier after plating, we expose them to sevoflurane 1.6% or dexmedetomidine. And the dexmedetomidine is always nowadays given along with other inhalation anesthetics to mitigate anesthetic-induced toxicity. And we have several papers published to show that it actually acts as a neuroprotective agent. And then what we do is that we do live diet assay. We use ethium homodimer 1, and those cells that are dead, they pick up the dye, they light up because it really is internalized only in dead cells. Then the number 2, we look at the neurite outgrowth. We stain them with neurofilament, and then the software analyzes the extent of growth. And then we also look at synaptophysin at the presynaptic cell, and we look at the postsynaptic juxtaposition with PSD95. When these two proteins come together, we designate it as the synaptic connections. And then we also, we do as to where these anesthetics may be acting, we do mitochondrial morphology. We look at fused mitochondria, which is healthy, intermediate, or fragmented. And what we have shown is that in many cases, when the pups or neurons are exposed to anesthetics, we find oftentimes fragmented mitochondria, which then, we then determine ROS production because it produces free radicals, and which in turn causes really the neurotoxic effect. So long-term brain cell recordings we have a special chamber whereby we keep these cells alive for days to week, and it has oxygen, PCO2, all other parameters as the brain cells were growing in the intact brain. We have large networks of these neurons which we can monitor directly by using either voltage sensitive dyes, or we can use calcium indicator dyes. And these cells could be monitored at the larger extent. We also can pair these cells directly on a semiconductor chip and our chips to be able to monitor their activity, how anesthetic shut down this activity, what happens early stages of anesthetic exposure, and what happens in the longer run when it is all washed out. We can also isolate brain slices because cell culture still is artificial. Some people say that, this is not how brain cells are connected. You're just randomly putting them together. We can use in the intact brain slice and then expose them to anesthetics and see the effects as to which particular part of the brain is more vulnerable to anesthetic. So the question is that inhalation anesthetic, does exposure affect neuronal viability, outgrowth, synaptic connectivity in cortical and hippocampal neurons? And what we actually found was that in the control situation, we do not see much of the dye picked up by dying cells, whereas in sevoflurane, as well as in desflurane causes more cell death than what you see in sevoflurane. So my preferred choice will still be sevoflurane. And you can see that a lot of these dead cells are present once they were exposed to desflurane, lesser number in sevoflurane, and almost no cell death occurs under control condition. So when you merge these images, you can actually see, with neurofilament staining with, the dye of living cells, and you can see a large number of dead cells here, lesser here, and almost no cells that die under control conditions. So this is really the viability assay. We find that sevoflurane doesn't cause much of cell death, but desflurane significantly reduces the number of plated neurons. And then we looked at the extent of the growth, which we can really measure quite accurately. And we find that in the control conditions, the cells have extensive growth, but in sevoflurane, we see some curtailment of growth, but desflurane once again suppresses growth quite significantly as compared to these control cells that are not exposed to anesthetic. They're only exposed to air. So the growth quantification, again, we see that the sevoflurane and the desflurane, both of them suppress the growth of the neurite, the extent of the length of the neurite. So meaning that the brain cells wouldn't be able to reach too far to connect with its partners. And so the time course was also interesting. If we looked at really after 24 hours, it was surprising finding that the desflurane exposed brain cells actually had initially started out really fast, but then they tapered, and then eventually after 8 days, both sevoflurane and desflurane resulted in a suppression of the extent of growth of brain cells. And then we also looked at the expression of synaptic proteins. Under normal conditions, what you will see is that, if you're looking at the presynaptic marker, which is synaptophysin, and the postsynaptic marker, which is PSD95, we find that both this is in the control, but sevoflurane reduces the total number of synaptic connections between neurons. And desflurane does the same thing. So both of these anesthetic agents, they reduce the number of synaptic connections. And then also we looked at really mitochondrial morphology. We find that mitochondria are very happy and healthy, and in the fission state, they are really all pulling together like a train and providing energy, moving the cellular functions forward. Sevoflurane, they become fragmented, and then more fragmentation is seen in the presence of desflurane. So what we think is that the anesthetic induced effects maybe on cell death, maybe on growth and synaptic connectivity have something to do with anesthetic's effect on mitochondrial function. And this is really, we then used a mitochondrial division inhibitor to see whether this is really the case. And when we use MDV1, we find that it acts as a neuroprotector. So even in sevoflurane and desflurane, what we will find is that in its presence, the mitochondria stay healthy and happy as compared to what you would normally see without it. So MDV1 also restores the extents of growth. So it is, again, protecting mitochondria, will protect cell death, will protect anesthetic-induced perturbation of growth, and it will also protect the number of synaptic connections. So we also asked whether the acute exposure of cortical or hippocampal neurons to inhalation anesthetics, what we do. So we have published this, it's already published. We think that at the anesthetics effect at multiple levels, we have ROS-mediated DNA damage, we have excessive endoplasmic reticulum calcium release, which causes calcium-induced cytotoxicity. And then in the neurites, you have mitochondrial-mediated RAS and caspase production, which is neurotoxic. So the idea really is that it will eventually produce the hallmarks of what you see in Alzheimer's disease, where beta amyloid and tau plaque, they accumulate and eventually compromise neuronal communication. So this is really the takeaway message. The summary is the sevoflurane and desflurane, they inhibit outgrowth and cause neuronal damage and premature neuronal culture. Sevoflurane and desflurane, they compromise the expression of synaptic protein, leading to network dysfunction and deficit in the network. And then both of them mediated cytotoxicity likely involves perturbation of mitochondrial structure and function. So this is really the takeaway. We then asked the question, what happens if you take the pregnant female or a newborn rat pups and expose them to anesthetic? What will happen to their learning and memory when they are adult animals? So what we do is we could use subcutaneous dexmedetomidine. We then it's kind of as a preventive measure in these pups. to make sure that it will protect them, or use controls where we just inject control solution, expose them to sevoflurane either 1.6% or 3.2%. Both are clinically relevant. And then we wait until they become P60 pups, which is young adults, and then we do behavioral testing. And behavioral testing, we do Morris water maze, where the animal has to really look at, find the platform where it's hidden somewhere. And then we do second is a novel object recognition or test whereby we habituate the animal for 15 minutes for 2 days. They become familiarized. Then we ask them to find the object that whether they have now retention of that particular novel object. And this is how they're tested. The third test we do is that open field testing, where the animals are habituated with their environment. Then we give 3 shocks and then ask the question on day whether they still have fear conditioning or not. The interesting thing, it turned out that even though in Morris water maze, the animals could find this platform in novel object recognition, they passed to. But the 2 important things was that animals that were exposed to sevoflurane 3.2%, they had a hard time really initiating that movement towards the target. And the other thing was, even though they found their platform, but they were spinning so fast in an ADHD hyperactivity mode that it took them, when you did the heat map, it took them the same time, but it was at the expense of random stochastic search for the target. And so we then ask the questions, if we expose, we do the heat maps. I won't bore you with this. The animals, for example, tend to really stay where they are. They were hesitant and or also really had more random stochastic behavior after sevoflurane exposure. And finally, we really asked what happens to those neonates that are exposed earlier to anesthetics. And when we test their learning and memory, after learning and memory, we take their brain out. And also the young pups, when we expose them, we also extract their brain. And then we do PCR to look for differentially or nanostring, in this case, the differential expression of various genes. And we find that the animals that were exposed to anesthetic had several genes that were up and several down regulated. And most of those genes that we found up and down regulated, they control many important functions, which is the cell survival, circadian rhythm, axonal migration, learning and memory, long-term neuronal plasticity. So most of these genes that we discovered were down or up regulated, controlled very important developmental aspect of brain cell connectivity leading up to synaptic transmission. So we are teasing this apart right now. I think I am running out of time, so I will stop here and take any questions that you might have.
Thank you, Naweed. Thank you so much. Extraordinary talk, pure research, thoroughbred research. Very interesting. I mean, I was doing research before we started the conversation. Now, what is the trade name of sevoflurane? Is it a common anesthetic agent?
Dr.
It
Naweed, may I
is
ask a
one.
question?
It is one of the most common anesthetics, and I think exclusively for children, because it has a very fast induction and quick recovery. And the other, which reduces the hospitalization time for these kids. Also, it doesn't irritate like halothane or isoflurane will irritate your throats and make you sick and vomit. It doesn't have any pungent taste of smell, and you can have any flavor, gulab jamun flavor or any flavor. So the children actually like and love this particular agent, and it is commonly used for almost all surgeries for young children now.
So does it cause mitochondrial dysfunction? Because the research is it causes mitochondrial dysfunction, apoptosis. You mentioned a few things, neuroinflammation, synaptic dysfunction. So by the look of it looks rather probably more damaging than relieving.
So
Yes.
we have really tested propofol and all of these agents. We find sevoflurane to be the least neurotoxic in that category. But the issue really is it's the catch-22. You cannot perform surgeries in wakeful animals, right? You can use maybe somebody's socks to put an anesthetize a person, or pads of our ice hockey goalie. They stink so much that half people, the other player, when they come to score, they pass out. So so the idea really is that you have no choice. So, understanding where they act and what the long-term effect might be, how could you then really come up with, the idea of, performing surgeries in most of these patients?
So lots of people want to ask questions. Very senior scholars sitting here, Dr. Mulazim Bukhari, Dr. Badali Ejaz, please go ahead. Sakina,
thank you
would
so
you
much.
allow them to ask questions
Yeah,
directly?
there are a couple of questions on the chat.
Okay.
Can we cover those just so we don't miss them? So
Okay.
I think Dr. Habib Shoaib, he said, I faced examples of babies who have behavioral problem. Their mothers give a history of gestational bad conditions. That's more of a comment, I think, than a question. And then Dr. Ajaz, he had a question. Do you want to ask the question, Dr. Ajaz?
I have a question, madam.
Okay, go ahead.
Okay, I'm Dr. Ali. I'm practicing anesthetist in Rawalpindi. So we have a lot of neonatal and pediatric population. I'm anesthetizing those on daily basis, and a lot of children with multiple surgeries. I haven't heard this kind of complaint from the parents. Sir Naweed, can you please put light on the issue that how much exposure practically causes this mitochondrial development and neuronal development issues?
I think it's a great question, and it's really an ongoing debate. What I see in our children's hospital, I was the scientific director for Alberta Children's Hospital. It's one of the largest and one of the really best hospitals in Western Canada, where I see these children who are coming for circumcision, and they are completely knocked out. In our countries, we don't really anesthetize these kids. We maybe give the local anesthetic, but they're completely anesthetized. And, so we argue that this is completely unnecessary, but they don't really want to take any risks. But the issue that you have brought up is the problem is, well, in some cases, luckily, synaptic plasticity that will help, maybe repair or recover some of the damages that are caused by anesthetics, even in the mitochondrial function. So parents will never come back to you because you never follow them. And we also see these neonates, premature babies, when they are born, they will have cognitive dysfunction, right? But there's nothing you could really do in that particular case. Our really idea is that the learning and memory, these children will be affected. And of course, no health agency, no doctor or anesthetist will say to the patients that we are anesthetizing, but this may happen to your child. You have to really do something to improve their cognitive function. So I think what we ended up doing is that we have created a Worco Child Development Center, whereby we know that these children will have some perturbation or behavioral alterations. We provide them with enrichment strategies in that particular cases. For example, the language. For example, we could provide them, some interactive social integrations, maybe some physical activities to make sure that if anesthetics really affected them, their brain connectivity, that you could mitigate their effects. But for example, in your case, you're doing everything which is right, and because you cannot perform surgery because pain itself has been shown to exacerbate the similar types of brain connectivity patterns. But I think the lesser we expose them, normally people will do maybe 30-minute surgery, but if you have congenital heart problem whereby a child has to really go through open heart surgery to fix some of the valves and other functions, those kids are down for several hours. And in some cases, the children also end up having multiple surgeries over a very short time period as well. So I think all of these effects, somehow maybe we can collaborate with you. I don't know I don't know if you're associated with Pindi Medical College, Dr. Omer. I actually inaugurated their research center before COVID and give a talk there too. So we have set up a collaboration with them to find really means that we could put our resources together and then follow those children, because the children are the asset for any community, and we don't want them to be affected through any of these agents when the effects could be mitigated.
Doctor, I have a
There is more input related to this. Um, I'm working here in eye hospital, and we're doing surgeries for retinopathy of prematurity. Those are the premature born babies, and we are doing them totally on total anesthesia is maintained on inhalational anesthetics, specifically isoflurane and sevoflurane. I want to ask if there is any data on isoflurane, or what impact it carries on the premature babies.
So I think, again, the brain is developing at the speed of there are 30,000 synaptic connections that are established in a developing brain, even in the neonates, much, much faster. And the other issue with neonates is that if you look at many of the proteins that you find, for example, in Alzheimer patients, phosphorylated forms of tau, it is 5 times elevated than a normal adult who has the age of 18 years to 70. And they are AD patients, people. So newborn also will have tau-related proteins 5 times upregulated. So these proteins are playing really very important role in also connectivity and recognition of brain cells. But sevoflurane has been found to be, by us, least neurotoxic, whereas isoflurane or halothane are absolutely deadly. And here in North America, I do not believe anybody uses them anymore. So it's the sevoflurane and desflurane, and they are always given together with dexmedetomidine. So dexmedetomidine is now commonly used as an adjuvant to prevent and protect the neurotoxic effects that could be induced by any inhalation anesthetic.
Dr. Naweed. This is Dr. Madad Ali.
Ji, Assalamu alaikum, Madad bhai.
Assalamu alaikum, kaisan hai? You gave an excellent, thank you. You gave an excellent talk. I really enjoyed it. And I have a question. We are using a lot of propofol in endoscopy. Every day is being used propofol. Is there any study on the propofol effects on the neuron?
I think now what happens here in our hospitals, even for when people are given sevoflurane for general anesthesia, they always include propofol. And what nurses tell us that the children, when they recover, they are less agitated and they are more calm. And then they also do not complain of the pain that is inflicted by surgery. But I think we have published some papers with propofol too, but because it's really not that extensively used over a longer time period, like a general anesthesia is maintained, because I think there are many ethical issues also when you are using agents such as the volatile anesthetics, because they get into the atmosphere and they really destroy the ozone layer. So people have to be very careful. Another anecdote, it's very true in North America. I don't know how true it is in Pakistan. Most of these anesthetists who function in a hospital setting, they end up having female children as opposed to male children. So even if they have male children and they have multiple children, the larger number will be female. And we don't know what it is that the annihilation anesthetics are also doing to your chromosome at the chromosomal level. But I think for propofol, it is effective, it works, and it gets cleared very, very quickly. It is, I think, also a choice here as well.
Any neurotoxicity from propofol?
We did show actually that it does more or less very similar kinds of things that you see with other anesthetics. We have published a couple of papers on propofol. I'll be happy to send you those.
Thank you very much. I appreciate it.
There is a question on chat from Dr. Ajaz Haider. Is there any evidence of general anesthetic exposure in pregnancy or during childbirth and the risk of autism in children? Have there been any trials comparing, example, cesarean sections under general versus spinal anesthetic?
So actually, this paper was published just this week in Nature, and they compare and contrasted the differences between vaginal birth as in the cesarean birth. And they found that the children who are born through the vaginal normal birth, they go through a whole, a large number of bacteria that are maternal, and they will influence the baby in a very constructive and positive way as opposed to cesarean. I think you can check that out, and I'd be happy to share that post. So they compared and contrasted the differences between vaginal birth and the cesarean birth. But no one, I think, has followed up on those children where pregnant mother, late stages of gestation, when she was exposed to anesthetics for any general surgery, spine or other required bone repair or other things, what happens to the newborn? Because it does crosses the blood placenta as well as the blood-brain barrier to have its effect. But nobody has actually followed up on those studies. But because of the precaution, our Canada Health has now required that all anesthetics, when they are given to pregnant mother or newborn or young children, that they be warning labels be placed on those. But the dealing with these neonates and the newborn is a bit tricky because we don't know, for example, when a baby, a newborn, in even a neonate, the born prematurely, is exposed to painkillers. Sometimes they give morphine to manage their pain. We don't know if the actual receptors for these particular agents are yet developed in their brain or not. And if they are, do they get sensitized or desensitized? Those studies have not been done, and I think it is about time that somebody really looks at those. But the link to autism spectrum will be quite subjective because there are at least 200 different genes that are altered in those children in their brain, which are irrespective or irrelevant of any other exposure, for example. I mean, the anesthetics may exacerbate because in autistic children, it's the balance between excitation and inhibition that is altered, that is perturbed. So if anesthetics during early stages, when the most connections in the brain are GABAergic, and GABA is excitatory, and if anesthetics really augment this particular communication, synaptic transmission between those brain cells, I wouldn't be surprised that the hyperactivity or ADHD that you see, or obsessive compulsive behaviors that you see, or even the autistic traits that you see, whereby the actual connectivity is shifted from maybe social aspects, cognition, towards more IQ facilitation. So that work still, I think, needs to be done.
So ADHD, is it a lateral spectrum of the autism or is it something different?
I think it's a different, we just published, Shabih bhai, a paper, very extensive paper, a review article. I'll be happy to send it to you on ADHD in younger children versus the adult, because I think most of us are also have some ADHD traits, which gets us, to get by. But for those children, they
I'm
are.
one of them.
No, you're not. I think you're the smartest one among us. But so we have deciphered and looked at the role of dopamine in those particular pathways that really are. And there are medications which we debated whether mothers who have ADHD, and when they are given those drugs, stimulants and non-stimulants, what will happen to their offspring. So one of my PhD student, a PhD MD student, is looking at exactly at that aspect, where he has these pregnant mothers, he exposed them to those compounds or drugs that are given for ADHD, which is very commonly and is becoming really very prevalent. What might happen to the newborn pups in terms of its learning and memory capabilities?
Thank you. I see Dr. Hassan Zaidi from India is a very senior surgeon. Would you like to share your experiences, Dr. Hassan Zaidi? If his mobile is working. Maybe not.
Naweed, Assalamu alaikum.
Assalamu alaikum. First of all, I owe you a huge debt of gratitude. We have, mashallah, Gharbi Sahab coming next week.
Yes, I know.
He accepted our offer. May Allah bless him. And I think we're so excited to have you. And I think I owe you a huge debt of gratitude, maybe a box of gulab jamun.
You're welcome. My question and my comment and observation is things which have been taught from time immemorial. Naweed, you said that the babies who were born from vaginal deliveries had, by virtue of having an atmosphere with the mothers, the bacteria, which are helpful. The studies which I was taught was that babies born through cesarean sections were very much intelligent as compared to babies who were born through the normal passage. What is your opinion about that now?
I think it's really a great question. Back then, we didn't really know much about gut microbiome. And we know that these gut microbiome that determines your metabolism, which in turn really influences metabolites, which are then connected to our connectivity, brain connectivity, and behavior as well. And we know that, many neurotransmitter, for example, serotonin, 80% of it is synthesized in our GI tract and in our sympathetic and parasympathetic nervous system. So the recent paper, they really looked at many different parameters that the actual vaginal birth, when the baby was exposed to mother's bacteria and other environmental factors that are normally in the tract, that these babies had better immune system, they had fewer allergies, these babies also were healthier, but I don't think they looked at the cognitive aspects of it. So their sensory perception in the immune system and their oxytocin levels and the connectivity with mother was far more greater than children or babies who were born through cesarean. So, cesarean births are really not a trend, but it's a necessity because, the baby is in distress. So you have to really take them out. And so, I think in some cases they also do cesarean because there could be cranial damage when the baby is going through the birth canal. So I think the study, if you just did a search, I think Guardian also published an article on this as well, that those children who are born through the natural process of birth are better in terms of their immunity, I don't think they really, I don't remember if they looked at the cognition because the study is just very recent.
Okay. Thank you very much, Naweed. Your lecture was very, very interesting, full of knowledge for an old man like me. And
You're not
I
old.
congratulate you for all the researches and you're doing. Excellent. I hope we meet soon. I'm right now in India for a wedding. Otherwise, I would have joined Maayatullah for your kind invitation. Thank you very much.
Yeah, you will always have home here, and I think it's about time, Hassan Bhai, that you come and visit us, and we will give you a
Inshallah.
tour. And we will also show you a robot that goes inside the magnet to perform the brain surgery.
Okay, thank you so much. Thank you.
Dr. Mulazim Bukhari had raised, he was there to ask a question, has disappeared. Are you there, Dr. Bukhari, anywhere?
I don't see him. I'm
Over to
sorry.
you,
Yeah.
Mulazim.
Yeah.
Assalamualaikum. Can I ask a question?
Of course.
डॉक्टर नवीद मेरा नाम शुजात है मैं यहाँ पे एज यूके में एज ट्रस्ट रजिस्ट्रार काम कर रहा हूँ एज ए बीइंग अ पीडियाट्रिशियन सो बहुत ज़बरदस्त आपकी टॉक थी मुझे सिर्फ दो और मैंने जो क्वेश्चन दो फॉर्मुलेट करे हैं वो एक्चुअली मुझे उसका जवाब जो दीगर कोलीग ने सवाल पूछे उसमें मिल गया है बट आई जस्ट वांट कि आप थोड़ा सा मुझे क्विकली अगर गाइड कर सकें मेरा
Hassan
पहला
Bhai, if you
क्वेश्चन
could, I can't actually hear you. Okay, go ahead now.
या सो मेरा पहला क्वेश्चन जो मैंने आपके जो लेक्चर से मैंने एक कॉन्सेप्ट मेरा डेवलप हो रहा था कि exposure time is very important, especially like neonates, which are these babies have a developing brain, so they are more vulnerable to exposure अगर उनका होता है तो sevoflurane. And then again the older child who has got a developed brain, so again in developed brain में हो सकता है कि exposure time will be more accountable for the damage. So इसके ऊपर has there been any study that where the different stages of the developing brains have been studied with respect to the time of exposure?
Yeah, so I think it's a great question. Our brain continues to really develop until the age of 25, right? Your spinal cord starts developing by the age of 4 or 5, but your brain continues to develop. So when you think about development, when a baby is born, baby would have twice the number of brain cells than he or she will end up after birth, a few months into the birth. So a huge number of brain cells, half of them are killed by natural programmed cell death. And then the connectivity really starts from there onwards. And so they will connect, they will talk to each other. And you can see that notwithstanding the fact that half of the brain cells are killed, the brain grows quite significantly. This is all synaptic connectivity. And it really, it's just like a popcorn popping in a popper. And we don't know which particular area of the brain will be affected. So I think all of these stages until 25, the brain is vulnerable to injury, trauma, abuse, neglect, all of those aspects. But the younger the child is, the more vulnerable their brain is because the connectivity, the cells haven't even reached their potential target. Or in the case of hippocampus, where learning and memory occurs, the myelination actually doesn't occur until 7 to 12 days after birth. And this myelination plays an important role for rapid communication between various parts of the brain. So I think all of these stages are critical, but the younger the child is, the more vulnerable he or she will be.
थैंक यू और डॉक्टर नवीद लास्ट क्वेश्चन जो ना मेरा रिसर्च से रिलेटेड है कि ऐसे आपने मेंशन करके देयर आर लॉट ऑफ अदर फैक्टर्स जो कि कंपाउंडिंग इफेक्ट्स क्रिएट करने लगते हैं जैसे कि निगलेक्ट है और इस वक्त यूके के अंदर चिल्ड्रंस मेंटल हेल्थ के हवाले से काफ़ी क्राइसिस है सो आई वाज जस्ट वंडरिंग कि अभी तक कहीं पे एक ऐसा जैसे कि फ्रेमिंगम हार्ट स्टडी थी जिसमें एक लॉन्गिट्यूडनल स्टडी की गई पॉपुलेशन को ओवर द पीरियड फॉलो करा गया और देन अ लॉट ऑफ लाइक कंपाउंडिंग फैक्टर्स मेरे ख्याल से उसके बाद क्लैरिफाई हुए कि विच आर एसोसिएटेड फैक्टर एंड विच आर द कॉजल फैक्टर सो क्या इस स्टडी को रखते हुए एंड कीपिंग इन द माइंड के mental health issues भी in countries में, western countries में काफ़ी different वजूहात से हो रहे होते हैं, जिसमें neglect abuse भी है and the exposure to the substance abuse. So कोई इस तरह की longitudinal studies या Framingham Heart Study की तरह से कहीं in western countries में है इस वक्त?
I think for anesthetics, people did do a study where they looked at twins, and they studied twins all across to compare and contrast if one child had to have surgery, the other had to go through anesthetics or not. So they tried to follow those kids, but they couldn't really find anything in that context. But I think a longitudinal study is really important, but you need to define the parameters that you will be looking for. Until and unless you do basic fundamental research, it will be very difficult to find as to what you're looking at. So just looking at needle in a haystack. So I think it's about time that those studies are looked at. And you talked about Pakistan and other countries. Pakistan में तो देखिए हमारी brain में इतनी resiliency develop हो जाती है कि वो brain is really resilient to any short-term shocks. Here we see in a children's hospital, most of the patients who are in chronic pain, they are young girls because their pain perception is really exacerbated by their interactions with their mothers. So हमारे यहाँ तो चोट लगती है तो वो जाए तो माँ फौरन प्यार करेगी, कहेगी go, you will live, कोई बात नहीं बेट. Here they catastrophize that particular aspects of pain. किसी ने उंगली भी रख दी तो they just jump and react. And I think mental health issues here are prevalent and prominent because of the actual diagnosis. In Pakistan and other places in third world countries in Africa, nobody really cares. They just tell these kids, go and knock yourself out, संभल जाओ. और उसमें mental health के भी depression, anxiety effects are always shadowed. So it's really not that it's a prevalence of mental health related issues in North America. It's actually the awareness and the diagnosis, whereas back home it's mostly the stigma that is attached. People don't want their children to be known to the community or doctors that are having mental health issues. So I think there are many confounding factors that need to be taken into any study that somebody will design both retro and proceptive.
उंगली के ऊपर एक बात हो गई before we conclude. मुश्ताक अहमद यूसुफी साहब बड़े मशहूर थे हमारे.
Can we keep the questions in English, please?
Well,
because there are people who may not understand Urdu. Sorry.
This talk has to be in Punjabi, unfortunately. Naweed will translate into English. Just 1 second only. So only Firdaus was a very famous dancer and singer in Pakistani film. She sings, and the dupatta touches the finger of the hero. So she says, "Oye, tusi to meri jaan hi le li."
So very true. And I think the same thing happened. I was forced to watch one of the Indian movies with Shah Rukh Khan and he was going on and on about the poetic sense. And she said, well, how come you don't say those things to me? And I said, you know how much he charges? He charges $4 million to just say that.
So apologies to you, Sakina, for that. Thank you. And Naweed, excellent talk as always. Please keep 25th of December free, because we're starting the Research Center in Ethics in Karbala during the conference. It's already inaugurated, but we'll have a full session in which you will be a speaker, inshallah. 25th of December in Karbala. That will be Al-Zahra University, inshallah. And over to Sakina for thanking and for the announcement. Thank you again, once again, Naweed, as always. Phenomenal.
At your service.
Thank you, everyone, for joining. We really appreciate it. This will be our last webinar for the year. We have a workshop coming up, but only for the people who have already registered. I will be posting a list of webinars for next year, inshallah, very soon. And hope to see you all again. Have a wonderful New Year's. Inshallah, we'll start fresh again in 2026. And thank you so much for Dr. Naweed. I really, really appreciate you and your knowledge and your humor and shairi. All of it. Thank you so much.
Thank you.
Anybody have
Thank
any
you.
questions, comments before we log out? Thank you.
Thank you, Naveed.